Hello, everyone, and welcome to our webinar today on unlocking the power of analytical ultracentrifugation. Today, our presenter will be Dr. Borries Demeler, who is a Canada 150 Research Chair at the University of Lethbridge with cross appointments at the University of Texas Medical School and the University of Montana. He's renowned for his contributions in solution biophysics, particularly in the hydrodynamic modeling of AUC, and is a principal author of UltraScan. He also manages the Canadian Centre for Hydrodynamics, where his recent projects include characterizing lipid nanoparticle formulations, viral vectors, and structural functional studies for the SARS-CoV-2 viral proteins. And today he will be presenting on the foundations of AUC and highlighting some of the research that he's done in the area of AUC. So I will pass it on to our presentation then. Thank you.
It is my pleasure to present today's seminar, sponsored by Beckman Coulter Life Sciences, on unlocking the power of analytical ultracentrifugation, or AUC. I'll start off with a disclosure that Beckman asked me to present, which is shown on this slide. I'll begin with an overview of analytical ultracentrifugation by covering the background and the hydrodynamic fundamentals. Then I'll discuss a new approach for measuring analytical ultracentrifugation experiments called multi-wavelengths AUC, and discuss some application examples. Starting off with AUC background, what is AUC? It's a solution-based separation technique which is used to characterize molecules on the nanoscale. So we're talking typically anywhere between 1 to 100 nanometers. In that range, molecules can be characterized by AUC. The information that we get includes the hydrodynamic radius of the molecules, their mass, globularity, which is a measure of shape, as well as the density of the particles. We learn also about interactions between molecules and can measure their strength of interaction as well as slow kinetics. AUC is a first principle measurement technique that does not require references, provided the instrument is working properly, which we confirm by using molecular standards and calibrations that assure us that the instrument is working. So unlike gel electrophoresis or size exclusion chromatography or mass photometry, you do not need a reference to make sure that your sample is actually measured correctly. The metrics from analytical ultracentrifugation come from the observables that we can model precisely with mathematical models and retrieve those molecular parameters such as sedimentation and diffusion and partial concentration directly from the instrument. So we do not need any references. The technique is widely used in the biomedical field and material science applications to characterize molecules on the nanoscale, and different optical systems are used to detect and characterize virtually any colloidal molecule that you can imagine. It is essential for biopharma to characterize injectable and liquid pharmaceuticals. So what can we learn from AUC? Very importantly to biochemists and other scientists working in the life science sector, the molecules can be studied in a physiological environment. That means the solution conditions can be adjusted to cover the similarity of, let's say, cytosol concentrations of ionic strength, pH, buffer type. Other molecules that may act as ligands can be added. The oxidation state can be adjusted by adding a reductant to your samples. You can modulate the temperature. And of course, you can modulate the concentration of the analyte that you're studying. So you can make the solution as physiological as you want. Furthermore, AUC can address a very large size range, which starts just with the size of a, let's say, a salt ion that you can sediment into a gradient, and you can measure that. So that would be about 100 Dalton range in the hundreds of Daltons. But we can go up to 10 to the 8th Daltons, which would be molecules as large as chromatin assemblies, as well as viral particles and large nanoparticles that are based on metals and are very dense. And the way we can achieve this dynamic range is by modulating the rotor speed. We can measure dynamics and interactions between molecules, which will give us access to the polymerization and molecular growth of self or hetero associations. We can characterize ligand binding of small molecules, slow kinetics, and the KD, the equilibrium constant of the interaction. We can use AUC to perform composition analysis, where we characterize the purity, the number of components in a mixture, the partial concentration of each analyte in the mixture, the molecular weights, densities, and globularity. The conformational analysis is further possible to characterize folding or melting studies of biopolymers, conformational changes based on changes in solution properties, and so forth. Now, people always have lists about things what you need to remember about a particular technique, and I have 9 things that I want you to remember. First of all, what do we measure? AUC only measures 3 things: the sedimentation coefficient, the diffusion coefficient, and the concentration of an analyte. The information that we are getting can be obtained because using centrifugal force, AUC will separate the molecules in the solution based on their size, their anisotropy, and their density. If you're not familiar with anisotropy, you can think about globularity. Globularity means the more globular a molecule is, the faster it will sediment and the faster it will diffuse. The more extended it is, the higher its anisotropy, and the slower it will diffuse and sediment, given the molar mass and density stays the same. The density can, of course, be characterized because molecules with a high density sediment faster than molecules with a small density. And that is a similar proportionality with the size of the molecule. Finally, how do we detect the molecules? There are 3 different optical systems in use right now. One of them is UV visible absorbance. So if your molecule absorbs light anywhere between 200 or 800 nanometers, we can then dial in that wavelengths where it absorbs, and we can measure this. Because of the fast detection in UV visible absorbance available with the Optima AUC, this can also be done in multi-wavelengths mode, where you can capture multiple or absorbances in multiple wavelengths ranges on that instrument during the experiment. Next, you can measure molecules based on their refractive index difference to a buffer, which can be done with Rayleigh interference optics that you can get with the Optima. And then finally, there's also a fluorescence detector that has been developed for the proteome lab, the predecessor for the Optima AUC, which will measure fluorescence emission of a fluorophore. So what can we then characterize with this technique? The short answer is anything that is in colloidal form. So that means being dissolved in a liquid. Among those molecules are biopolymers, proteins, nucleic acids, lipids, carbohydrates, or nanoparticles. Lipid nanoparticles are very popular. You may have heard about COVID vaccines, which are lipid nanoparticles that encapsulate messenger RNA, and there are many other uses for LNPs for vaccines. Silicon nanoparticles, synthetic polymers, metal nanoparticles, carbon nanotubes, quantum dots, and so forth. There are lots of different examples for that. What types of questions can we then answer? So for composition, we can then determine whether the molecule is pure or whether there are contaminants present. whether there is aggregation occurring or degradation, which is especially important for biomolecules. And we can do particle sizing and say how large the molecules in our mixture are. Next, we can tell about thermodynamics of a reaction, which may be caused by mass action. That is, as you increase the concentration, you get larger oligomers. So we can then determine the oligomerization behavior of a molecule and determine its dissociation constant, its off rate, and we can then follow that as a function of temperature as well. On the interactions, we can then determine interactions between proteins and other proteins, as well as proteins with nucleic acids like RNA or DNA, and we can, or even nucleotides for that matter, and we can measure proteins interacting with nanoparticles or nucleic acids interacting with nanoparticles, and we can then follow their complex formation, their stoichiometries, ligand binding, and binding strength. We can then measure vector cargo loading as in viral vectors or lipid nanoparticle vaccines, and we can determine effects of changes in the solvent, such as pH, ionic strength, redox states, and small molecules. Conformational analysis will tell us information about intrinsically disordered proteins, the unfolding of molecules, changes in anisotropy, maybe due to buffer changes, as well as allosteric changes. The density of a molecule can be followed by measuring the partial specific volume distribution, which may change upon cargo loading in, for example, lipid nanoparticles. And we'll look at some of the examples that are relevant for that. So how does an experiment, an AUC experiment work? You start with a titanium rotor. This one here is a 4-hole version. There's also an 8-hole version. And in each hole goes a cell, which you can see right here. And this cell has 2 compartments where you can load a sample. There's also a counterbalance that can counterbalance that weight, or you can put additional cells in there. The sector-shaped compartment in the cell then holds your liquid. It's sealed off at the top and the bottom by a sapphire window to make sure that it doesn't leak out while you're spinning it. And you will have an air pocket at the top, then there's a meniscus, and then the solution column. And the molecules inside of that solution column will then sediment outward from the rotor axis and pellet or pile up here at the bottom of the cell. We then shine light through there, typically by UV visible absorbance or Rayleigh interference, or a laser beam that then excites a fluorophore. The light is then measured, that either is transmitted or emitted or refracted, and a concentration gradient is observed that then over time results in multiple boundaries whose speed of movement we can then measure to determine the sedimentation coefficient. The shape of the curve is also proportional to the diffusion of the molecule, and so we can also measure the diffusion coefficient from that. So when we run an experiment, we fill a cell with an analyte solution, which is initially uniformly distributed. And then as we're spinning up the rotor, we form a moving boundary as these molecules sediment towards the bottom and clear the area between the meniscus and that moving solvent front here, or solution front. This whole process is modeled by what's called the Lamb equation, which has a sedimentation and a diffusion term. And the change in concentration over time and radius is then observed and modeled by solving this equation using finite element methods. So this process then can be precisely modeled by this equation inside of that sector-shaped compartment and gives rise to the molecular observances that we have here for the sedimentation and diffusion coefficient. Here's an example of how this works. So the rotor accelerates, the molecules start moving down the cell, and they pile up at the bottom. and you end up with a series of scans that have been measured in the experiment, and that gives rise to an AUC data set. This is for a single wavelength. There, we will also discuss how that looks, what that looks like for a multi-wavelength experiment. From modeling this concentration curve that we just saw, we can then extract the sedimentation and diffusion coefficient of each component that's in the mixture. Once we have the diffusion coefficient, we can use this equation here to determine the frictional coefficient of the particle. Together with the frictional coefficient and the sedimentation coefficient, as well as the partial specific volume of the particle, we can then determine the molar mass of the particle. And once we have the molar mass and the partial specific volume, we can use this relationship to determine the volume of a single molecule. Once we have the volume of a single molecule, we can then plug it into the volume of a sphere equation to get the radius of that sphere. This is called the minimal sphere, or R0, which together with the Stokes-Einstein relationship in this equation can then determine what the minimal frictional coefficient of a molecule were if we could take that molecule and squeeze it into a sphere. And so that frictional coefficient, or F0, is going to be smaller than the frictional coefficient that we observe for the molecule that is in its original shape. And from that ratio of the frictional over the frictional coefficient over the minimal frictional coefficient, that ratio gives us then the anisotropy phi, which is a metric for the globularity of the molecule. So we can obtain shape dependence for our molecules. We can get size information, the molar mass and the hydrodynamic radius. We can then also get the anisotropy of the particle, and we can also have access to the density or the partial specific volume by designing special experiments that will then give us distributions for the density of the molecules in the mixture. So to better understand what anisotropy is, we parameterize our frictional ratio, of phi, the diffusion coefficient and the shape of the molecule. If the frictional ratio is equal to 1, that means our molecule is a sphere. And if it's any larger than 1, it would be an extended molecule that is non-spherical. And for most proteins, the frictional ratio that if the molecule is well folded, it would be about 1.2, 1.3. But if it's unfolded or it is rod-shaped or disordered, we get values that are as large as 2.5 for the frictional ratio. If we have DNA molecules and fibrils or aggregates of linear molecules, we might see frictional ratios that are much larger than 2.5, and they would be larger than 3. It is important to understand that the shape of the molecule cannot be determined from analytical ultracentrifugation. All you can determine is the anisotropy or the frictional ratio. But it is quite possible that there is an infinite number of different shapes that have the same anisotropy, the same frictional ratio. So here, just for schematics, we have a dumbbell and we have a cylinder, and both of these molecules may have the same anisotropy. So we cannot tell by AUC whether one of them is a cylinder or a dumbbell in this case. So that's important to be aware of that we cannot tell different shapes, but we can tell different globularities of molecules. So in summary, we have an analytical ultracentrifugation trace that we can then follow over time. And we can then determine how many components are in that set of boundaries here that we've collected. So in this case, you can see that there's something fast moving up here where the distance between the boundaries is far, while down here, the distances are close together, meaning this is moving much slower than what's going on up here. Plus, we have a couple other boundaries here in between. And excuse me, the question then comes up, how many components do we have in our mixture, and what are their sizes and molar weights, and what are their anisotropies, their partial concentrations, and do these components interact? And if they do, how fast and how strongly can they do that? And then finally, we also want to determine the reliability of our measurement, which is something that we can answer by doing more extensive modeling of our data. So once we have our data, we can construct a two-dimensional plot. Typically, we plot the shape or the frictional ratio on the y-axis and the size, either as a sedimentation or molar mass range on the x-axis. And then any spot in that two-dimensional domain describes a different molecule. So for example, if our molecule sediments slowly and diffuses rapidly, we have lines that are very close together and that spread out very rapidly due to the fast diffusion. And those molecules would be found in the lower left of this graph. In contrast to that, we could have molecules that are very extended with a very small diffusion coefficient, and also fast sedimentation because they have a high molar mass. And so the size would be big, but the diffusion would be slow. And that would be found in this regime in the upper right part of the plot, where the friction is high and the frictional ratio is high, and the molar mass is large and the size is large. So there we would see these kinds of boundaries that are far apart, and they wouldn't diffuse very quickly and become shallow like here. They would stay more steep. And then, of course, we can have all kinds of things in between. And the task that we have is to find out which type of boundary is present in our mixture, and then how much of each do we have. So the relative amount of each spot in this plot is what we're after. In a multi-wavelengths experiment, we measure the same experiment at different wavelengths. In the same experiment, we just change the wavelengths and scan. And as you just saw, these wavelengths will have a different response that is proportional to the amount of absorbance that we see from our molecule at each one of these wavelengths that we're measuring. The wavelengths that we're measuring then provide a spectral information on the chromophores that are in the molecules that we're sedimenting. And these, the spectral information is going to be unique to each unique chemical component in the mixture. By analyzing this information, multi-wavelengths analytical ultracentrifugation provides then spectral separation in addition to hydrodynamic separation. And both dimensions can be analyzed and utilized to interpret the underlying system properties. We also can gain from the multi-wavelengths experiments by performing signal averaging, that is, by having signals from multiple wavelengths that we can then signal average to further improve the signal to noise ratio. So what can multi-wavelengths analytical ultracentrifugation then be used for? We have unique spectra from different chemicals that are mixed together in a solution, and we can then exploit that in our multi-wavelengths analysis by characterizing, for example, proteins that have different ratios of aromatic side chains to the backbone of the protein, where we can observe the amide backbone absorbance between 240 down all the way to 210 nanometers. And that can be distinguished with the aromatic side chains like tryptophan or tyrosine that absorb around 280 nanometers. Nucleic acids also have a very different absorbance spectrum from proteins. So if you have mixtures between proteins and nucleic acids, we can tell them apart spectrally. They just have different colors, and we can then tell how much of each color we have. And we can then distinguish fluorescently labeled molecules or fusion proteins that are fused to a fluorescent protein like GFP, RFP, or YFP, or any one of the other ones that absorb at different wavelengths. And the same is true for molecules that are labeled with Alexa fluors, which come in a great variety of excitation wavelengths that then can be measured with multi-wavelengths absorbance. Also, there are proteins that contain a heme or another cofactor or chromophore that absorbs, maybe indivisible, that we can then use to distinguish those proteins from other proteins that do not have these cofactors present. We can then also use multi-wavelengths detection to characterize the binding of small molecules. Many of the small organic molecules, like drugs, they have unique spectral properties in the UVs that can be distinguished from the absorbance that you get from a protein or another biomolecule. There are also quantum dots with size-dependent optical properties that we can then distinguish. Here is a single scan from a multi-wavelengths experiment. And what you can appreciate immediately is that there's a lot more information in a multi-wavelengths experiment because we got this second domain here that we're watching. That is the absorbance at different wavelengths. Here's the meniscus of our boundary and of our solution column, and here's the boundary that you can see. And this is a single scan. And over here, you see that the shape of these scans is very differently. They're very different. In this case, we have a DNA molecule that absorbs at 260 and has a peak at 260. But over here, we have a protein molecule that has absorbance at 280 nanometers. And then over here is the amide backbone absorbance at the bottom that we can also pick up below 250 nanometers. So isn't that amazing? From a single scan, you can already tell what kind of a chemical you're dealing with. Here it's DNA, and over here it's protein. Now, that information would never be available if you were just looking at a single two-dimensional scan. And that's the power of multi-wavelengths. We can then, of course, do numerical analysis of these mixtures of molecules that may have DNA and protein in them, and then tell how much of each we can do. So here's what it looks like when you have an entire experiment sedimenting, right? So these are multiple scans, and you get this moving mountain effect in 3D, where the boundary is basically sediment to the bottom, and you can observe this profile then over time. So how do we analyze this? Well, the way we analyze it is by slicing each radial position along the wavelengths domain to get a wavelength scan of the composition at that place. And that can be done for every radial position and for each time point in the experiment. If you make a mixture out of protein and DNA, you can see immediately that different ratios give you different absorbance profiles. So for example, in red, we have the DNA profile. In purple, we have BSA profile, which is a protein. And if you mix these at different ratios, you get everything in between. So the unique shape of this curve will tell us then something about the stoichiometry with which these molecules are mixed together. We can then take a slice of our radial profile and decompose it into the absorbance spectrum of DNA and the absorbance spectrum of BSA, and we can then tell the relative amount of protein or DNA is present at that particular radial position and reconstruct two new curves or experiments, one that is pure DNA and one that is pure protein. And if we look at different mixtures, we can then tell how much of each signal is present. So if you don't have any protein present and everything is DNA, all your signal shows up over here, in this case, a restriction digest of DNA. If, on the other hand, everything is protein, the picture would look like this. All of the protein is shown up over here, and there's no absorbance for the DNA. And then, of course, you get everything in between. When we analyze all of this, we end up with a two-dimensional plot in two different colors, in DNA for red and protein in blue, that then tells us where the molecules are in our mixture and what kind they are. So in this case, we know that everything occurring in blue must be a protein. So here we have the monomer, dimer, and trimer of our BSA molecule, while in red, we see the DNA fragments of our restriction digest of a plasmid, which generates 3 different fragments. And you can see that these fragments increase in anisotropy, or frictional ratio, the larger they get. So the 12th base pair fragment is relatively globular, although not nearly as globular as the BSA monomer. And a 208th base pair fragment is already more extended because, of course, it's longer. And then the 2811 base pair fragment not only is much more extended and has a higher frictional ratio than the 12 or 208 base pair one, but it also has more of a range that it covers because it's more flexible and it can have more conformations. And we see that reflected by the density of these curves here. In addition, we know that we have uncut plasmids and concatemers in the mixture that absorb at higher S values here. And all of this can be told apart with a multi-wavelengths analysis, which provides us very clean peaks here because we are, of course, signal averaging all the different wavelengths and globally analyzing it in this experiment. If we were to do this in a single experiment, let's say with the older instrument, the Proteome Lab XLA, this is what it would look like, and we wouldn't see much difference here. This is a single wavelength scan, and all you get is a bunch of peaks, and you can't tell which one is which. But in a multi-wavelength experiment, we can tell them all apart, even if they have very similar S values like these two molecules. They're completely separated. One of them is the protein, and one of them is the DNA fragment. And we can tell them all apart. So now let's look at some applications that can be used to be studied by analytical ultracentrifugation. Let's start with drug delivery systems like AAVs and LNPs, and then look at a sample for sample purity and oligomerization and aggregation are some other examples, which I will show. So adeno-associated virus, or AAV-based gene therapy, is a very important application in biopharma, where the essential question is, what is the degree of nucleic acid loading efficiency in your capsid? When you make AAVs, you always get undesirable side products that are either empty capsids or partially filled capsids, which need to be minimized. And we also don't want to have any free nucleic acids presence that will give us contaminants that are undesirable in the final pharmaceutical drug. And so we need to verify the loading state. And we can do this very well by doing multi-wavelengths AUC. And this was described in a paper by Amy Henriksen that you see cited here, which used multi-wavelengths analysis to determine the identity of various peaks that were then observed. In this case, we had peaks at about 60S. We also had a peak at about 20S, and we had a peak at around 100S, and we had a little bit of composition here in the middle. There wasn't enough wavelengths here to see exactly what this was, but because of the very clear picture here, we can tell that this is an empty capsid. It has the typical signature of a protein with a peak at 280, a valley at about 255, and then the amide backbone absorbing again at wavelengths lower than 250 nanometers. And that would match something that has this purple trace here, like BSA. For this peak over here, we have something that's more flat, like maybe like the green trace over here, which would be a mixture of protein and DNA. And that, of course, is our filled capsid, which has both absorbance from a protein and a DNA, and it looks much more flat. So when we analyze this data, we can then see that in the multi-wavelengths analysis, we have 90% of our material, or 85 to 90%, in the empty capsid form, the green being the protein signal and the red being the DNA signal. Then we have about 6% of the material being partially loaded, meaning it is containing both protein and DNA. And then we have about 8% of a filled capsid here, where the S values of the protein signal and the DNA signal overlap. They also overlap over here, where we have about 16% of the DNA co-migrating with the 86% of our empty capsid. This tells us that it's not truly an empty capsid, but that it is containing a little bit of DNA cargo, even in the empty one. And that gives a rise to about a 65 or 66 S species. In addition, we see that we have about 16% of a contaminant in there that is clearly a DNA contaminant, not a protein, because it just absorbs for the DNA. The protein signal does not have any signal down here. So we can, by doing this multi-wavelengths analysis, very clearly tell what's in our mixture. Another way that that can be done is by using what's called a analytical buoyant density equilibrium experiment, or ABDE, which can also be performed in multi-wavelengths mode. And we can then distinguish the presence of proteins and DNA in each peak. And what we see here is in blue, the protein, the capsid protein. So we have a big peak over here at about 6.5 centimeters with very little contribution from the DNA, but there's nevertheless some contribution from the DNA. And then over here, we have what looks like partially filled capsids that have more signal of protein than they have of DNA, meaning they're not completely filled. However, the filled capsid peak over here has an exact complement of protein and DNA, apologies, that is then reflected also in these higher density peaks that we see, which are possibly leaky capsids or aggregates that may produce additional density in these experiments by complexing DNA with cesium ions that are in the mixture, making the capsids even more dense than the filled capsid, which should be our highest density species, because the DNA gives rise to a more dense particle when they're compared to the one where there's just protein present. But we do get two additional peaks here, which are often seen when you measure molecules in cesium chloride. The next approach that I want to discuss is that of a lipid nanoparticle that's filled with RNA. And what we do there is a density matching experiment where we measure our lipid nanoparticles in 4 different ratios of light water and heavy water, D to O. And when you extrapolate the sedimentation coefficients that are observed in these different density data set, you get the density of the analyte at 0 S value. So you can extrapolate to S value equals 0, meaning that's where the molecule becomes equibuoyant with your solvent and where it will not sediment at all. So that's where the S value is 0. So you extrapolate to that point and you have the density of your analyte by taking the inverse of that partial specific volume value here. You get then the density of your component. When you do this for lipid nanoparticles, you will see that an empty lipid nanoparticle will float, and the more D to O you put in there, the more dense it becomes, the faster it will float. And you get these kinds of traces. You can do the same thing for loaded nanoparticles, and you can do this for molecules that are partially loaded, as shown here in the green. So here we see the trace that we get from a lipid nanoparticle that is not loaded. So this is very low density, having a partial specific volume above 1. The partially loaded ones, they have a nucleic acid phosphate versus amine for the cationic lipid ratio of 6:1, as well as the fully loaded particles that are having a ratio of 1. For each phosphate, there's one amine group from the cationic lipids. And you can see that when you have a lot more DNA in there, these become more dense, the partial specific volume will be reduced. And you can then characterize the density distribution that is present in these mixtures. Another example would be to characterize a monoclonal antibody. In such a case, it is important to know if there are any high molecular weight aggregates or contaminants present or degraded material. And what I'm showing here is an overlay of 4 independent experiments that were done at different concentrations, but using a wavelengths where the extinction is the same, so that we get roughly identical observance patterns and can easily compare them. Also, there are 2 batches of the same monoclonal antibody, and we wanted to know how well we can reproduce the compositional details of these samples. And as you can see, you can get very good reproducibility with the antibody oligomers being here visible at 8 and 10, and then the main peak at about 6.5 S. So that allows us to characterize the composition of a sample that is, maybe a monoclonal antibody vaccine that needs to be very pure and hopefully not aggregated when given to the patient. In the next example, I'm going to look at how we can perform these buoyant density gradient centrifugation experiments, the ABDE experiments. And what you're doing there is you're sedimenting a dense gradient material like cesium chloride or nicodens or matrizamide or iodixonol to establish this density gradient shown here in yellow. So the density of the analyte at the final distribution when we reach equilibrium will then determine what the density is at each point in the cell. Any molecule that may be also in this mixture here will then find a place where the density of the gradient forming material at equilibrium is equivalent to the density of the macromolecule. And the macromolecule will then sediment down to that point or float up from the bottom to also get to that point, and then it will be arrested and will be stationary. The peak that is then formed here is then characteristic for the density of the molecule. This kind of looks like this. So it's floating up from the bottom and sedimenting from the top until it reaches a point where the density of the molecule is identical to the density of the solvent at that point. And that's the trace that you see for a single component. This is represented by this equation up there, so we can precisely model this process and then figure out what the density is at that point. In Ultrascan, you can then characterize the density and get additional details about the identity of these peaks that are being formed by determining their density using calculations that are performed in Ultrascan. And we can then use them to characterize, for example, the loading state of a bacteriophage is shown here. And as you package more and more DNA into your capsid, you will get additional density that you can then measure by looking at the peak position that each species generates. So you can also have empty capsids as well as free DNA molecules. And depending on what gradient material you use, you get different hydration, which then also, is reflective of the solvent in which you're measuring, and it will determine where each one of these species goes in that final equilibrium gradient. So here's an example of the types of peaks that you can expect to see from a nicodens buoyant density equilibrium gradient or gradient equilibrium experiment that then gives peaks at different positions in the radial column here. And based on where that position is, you can then identify what the species is that is being sedimented here. And you can have mixtures, and you can get very high resolution between these. And that gives you a nice composition information about what possible capsid species may be present in your experiment. So this was a quick tour de force of an overview of analytical ultracentrifugation and how you can use the latest technology to characterize different experimental systems. And before I go on to answer any questions, I'd like to acknowledge our collaborators. First, I'd like to acknowledge Amy Hendrickson, who just obtained a PhD at the University of Lethbridge, performed most of the sedimentation velocity experiments and ABDE experiments that I showed here. Also, Saeed Mortazadeh, who together with Emery Brooks and Alexei Savelius from the University of Montana and Gary Gorbett from AUC Solutions, who have programmed all of Ultrascan. Then Kazuhiro Oka and Austin Seal at Baylor College of Medicine, who provided the AAV samples for the ABDE experiments. The AAV sample for the sedimentation velocity experiments came from Viviana Gradinaro's lab at Caltech. The samples were prepared by Zhao Ding and Tim Miles. At the University of Texas Health Science Center in San Antonio, Phil Silver's group provided the bacteriophage samples that I showed. And Aisha Demeler, my daughter, when she was at the University of Texas Health Science Center, performed the multi-wavelengths data analysis of the DNA and BSA mixtures. Now she is at the University of Lethbridge. And Helmut Köfen and Ingen Karabudak developed the multi-wavelength system that was used for the DNA and BSA mixtures that I showed. Thank you very much for your attention. I'm happy to answer any questions now.
Awesome. Thank you so much, Bo, for that presentation. So now we'll move into our Q&A section. And if you guys have any questions, if you want to pop them in the Q&A box down on your screen, I'll be able to ask them to Bo, and we'll hopefully get some good answers. So our first question then for you, Bo, would be, could you please explain a little further the differences between sedimentation velocity and analytical buoyant density equilibrium and the benefits of each?
Sure. Thanks. Thanks for the question, Amy. So sedimentation velocity observes the approach to equilibrium, which is what you're looking at in an ABDE experiment, the very end of the experiment, where everything becomes stationary. So when you're measuring in sedimentation velocity mode, you're also looking at the motion of the molecules through the solution column, which gives you access to the frictional parameters of the molecule. Once you're at equilibrium, the molecules no longer move, and so the friction goes out of the window. The reason why we use ABDE experiments where you can't see any friction is that we're able to separate molecules based on density alone. So the peaks that we see in an ABDE experiment do not reflect the molar mass of the molecule, at least not directly. They just reflect, the peak position reflects the density of the molecule. And so that's a very nice way of separating out the molar mass effect and only looking at the density, which is important when you look at, AAVs and LNPs as far as their loading goes. So the nice thing then about equilibrium is you can get rid of one of the variables that you don't have to model. Now, I should mention that the variable is actually still there because the width of the peak that you're seeing is proportional to the diffusion of the molecule. The smaller the molecule, the broader the peak will get. Hence, the diffusion isn't or the shape information isn't completely gone. It's just not as clearly visible as when you measure in velocity mode.
Awesome, thank you. And ABDE also requires less sample as well, if that's a sample constraint, which is one of the benefits of the buoyant density equilibrium. Okay, so the next one then, during your presentation, you talked about multi-wavelength and some of how you and Helmut worked together to try and like develop those. How have you seen that the addition of multi-wavelength has kind of changed or benefited the AUC researching community?
Yeah, I mean, the examples that I showed, I think were already quite convincing on the benefits of multi-wavelengths. It is just a way to add a separate or an additional separation dimension to your experiment. So instead of just separating by size and shape and density, now you can also separate by analyte type. So if you have different chromophores and different analytes, you can tell them apart by looking at their spectral properties. So it's adding another separation dimension to your experiment, which makes AUC even more powerful, right? It just gives you a lot more information. And by virtue of the ability to measure very rapidly, we also, in the new Optima AUC instrument, we also get a lot more data, which helps with the signal averaging, which helps with the quality of the data. So by adding more data, we're increasing the signal to noise ratio in the experiment. So our answers become clearer and they're more easily identifiable, the species that you have in your mixture.
Okay, thank you. And then along those lines, kind of, when you're doing multi-wavelength AUC, as you mentioned, you're able to look at different analytes within the sample then. So if you're looking at like protein DNA interactions, you'd be able to tell what those are. With AUC, is it also able then to look at interaction parameters and start to calculate K-off and KDs from AUC?
Certainly, that's absolutely possible. As you know, mass action is the approach by which we characterize oligomerization experiments. That is, by measuring molecules at different concentrations, we can modulate how much of the molecule is in, let's say, dimer form or in complex form versus free in solution as a monomer. And by measuring the concentrations, which is one of the parameters that you measure in AUC, you get very accurate representation of the equilibrium states with which these molecules interact. And from the ratios of these concentrations, you can then derive the KD. And if you're measuring fast enough, that is, your KD or your K-off rate is on the scale of the sedimentation velocity of the molecule, so which you typically can enhance by running at very high speed. You can then get some kinetic information as well, although I should caution that this is at very, at fairly long time scales. So probably 10 to the minus 3 per second, 10 to the minus 2 is probably the upper limit of what you'll ever see. So it's on the order of minutes and hours that you can measure in an AUC experiment for the K-off rate. So kinetics, not so much, but equilibrium constants for sure. That is bread and butter application for AUC to measure KDs.
Awesome, thank you. And then it looks like we maybe have one other question here, which was just what are the main benefits of using AUC compared to other techniques? If you want to maybe comment briefly on that. I know there's probably lots to go into that, but.
So every technique has its strengths and weaknesses, and the same is true for AUC. However, if you have the resources, then AUC has a unbeatable benefit of being first principle, meaning we can model the experiments precisely with mathematical equations and get absolute parameters out of the instrument that then allow us to characterize our molecules without the requirement for a standard. Everybody knows when you run a gel electrophoresis experiment, you got to put a molecular weight ladder there to which you can compare your molecules, because there's no analytical equation that will tell you exactly what the size of your molecule is based on how far it has moved in a particular buffer, in a particular gel, at a particular voltage, at a particular temperature. There's nothing there that relates all of those parameters together so that you can reliably tell how far your molecule has moved then, or what type of a molar mass is represented by a particular band. In an AUC experiment, you can do that, right? So that's the beauty of AUC. You don't need a reference standard to compare it to. The same is true for SEC. You run a molecule that's very extended. It goes much slower, and it looks like a dimer when in fact it's a monomer. But in AUC, you can get access to the diffusion coefficient and the frictional parameters, and you can tell that it's still a monomer even though it's very extended. And that clear information would never be available from an SEC experiment, for example, right? And for many other techniques, mass photometry included, you need reference standards to compare everything to, because there's no set of numerical equations that allow you to calculate the observables and figure out exactly what you got. So that's the unbeatable advantage for AUC. So if you have an AUC, you can get much more definitive answers for your samples without the need of a reference standard, for which you may not even be sure whether the reference standard is appropriate, right? In SEC, you might make a ladder out of a globular protein, but it just doesn't apply for an extended molecule. And so if you use the wrong reference standards, you still get the wrong answer, even though you have a reference standard. So that's the problem with reference standards. In AUC, you don't have to worry about that. That would be my biggest advantage of AUC. And then in addition, I would say that sample requirements are generally low enough that you can pretty much measure any molecule of interest using AUC without great expense, and you can obtain probably more information with AUC than pretty much any other technique out there, at least on the macro scale, right? If you want molecular structure, you have to go to different techniques that are also first principles, like SAX or X-ray crystallography or NMR, but those are much more involved. And with AUC, you get really nice results with not too much extensive modeling. That's all handled in software now and can be done very easily. So yeah, I think AUC is definitely something that everyone should be looking at.
Awesome. Thank you so much. And thank you for such a great presentation today. It was really enlightening. And I find every time, even though I've worked with you, I find every time you present, I learn more about AUC. So I really appreciate it. And then I just wanted to thank everyone else for taking their time today to listen to us and learn about some of the research that Bo's been working with and learn more about AUC. We'd like to thank LabRoots and Beckman Coulter for the sponsor of this. And then before we go, I would like to thank the audience for joining us today again and for the interesting questions that were asked throughout the presentation. If there were the questions that we didn't have time to get to today, and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand after we've finished the live broadcasting, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. So thank you again, and until next time, goodbye.